Hello students, once again welcome back to our biology class. Myself Dr. Raghavendra here. Well, in the earlier class we were discussing about what is HGP, Human Genome Project. Introduction we studied, right? And then what is the genome? Genome refers to be the total number of genes present in the haploid set of a chromosome. What is a gene? Gene is the part of DNA. Gene is the series of yes nucleotides or sequences of the bases, we can say. So every sequence of the DNA has its own meaning. The gene can be maybe around 3000 bases, or it may be 100 bases, it may be 200 bases sequence, it may be 1.4 million bases or 2.4 million bases. Many, yes, length it varies. So this kind of sequence has its own meaning to produce a particular kind of RNA, particular type of a protein. Yes, you might have been studied central dogma of the cell. What is that? DNA making its own copy of the DNA we called as a replication. DNA making RNA we call it transcription. RNA making protein, yes, we call it as a translation. So the entire central dogma of the cell depends on the type of DNA. That's why understanding the sequences of the DNA, its meaning, its use, its defect, its disease, all is very important, and that can be done by mainly Human Genome Project. Well, so in the earlier class we discussed about the goals of the Human Genome Project: to retrieve the information, to develop the database, to transfer the technology, to develop the tools, okay, to address the ethical, legal and social issues also which can arise by the yes output of mainly Human Genome Project we have discussed. In today's class, let us understand the history of Human Genome Project. Some of the points I have told you in the earlier class that 1970 there was a person, yes, Gilbert Walter, developed the electrophoresis technique that has become the basis for separation of the DNA based on their length or their molecular weight. And then, yes, in 1980s, the CEO of a company, Celera Genomics, his name was Craig Venter, proposed a proposal of Human Genome Project for decoding this DNA and understanding or bringing out the sequences information, and his idea was to sell it to the MNCs to make the medicines and pharmacogenomics, what we say. So information stored on the computer, mainly biological information, we say it as a bioinformatics. Along with the Human Genome Project, two important fields also arise, what they are called as mainly pharmacogenomics as well as bioinformatics. Well, so all that we have discussed. Let us understand in today's class the history of Human Genome Project. So HGP history as such. HGP started in 1990, right? So HGP started in 1990. Yes, initiated by US Department of Energy and National Institute of Health, NIH we say. So NIH refers to be the National Institute of Health of USA, in collaboration with the trust, what you can say, the Wellcome Trust developed by the UK, United Kingdoms, in collaboration with Wellcome Trust, which is a scientific committee of the UK, United Kingdom, okay, and further other nations joined in HGP as such: France, Germany, Japan and China. Yes. The HGP, yes, it has been, what you can say, thought it may extend for about 15 years. So earlier it has been mainly presumed, earlier it has been assumed that HGP may require 15 years to be completed. But so here, but with advancement of technology, but with advancement of technologies, HGP got completed in 1993, means 2 years before only. Yes, due to the advancement of technology such as mainly Frederick Sanger's method of automated sequencing, they have utilized gel electrophoresis, they have utilized DNA fingerprinting, they have utilized PCR, they have utilized number of techniques have been utilized mainly to complete this human genome project or the mega project. So thus it has been completed. Being a large chromosome 1 was the last. Chromosome 1 was the last to be sequenced. Okay, and completed in May 2006. Chromosome 1 has 2968 genes in it. That's why it has a more number of genes and sequencing took a bit more time. Otherwise, yes. So this is what we say regarding the brief history of mainly the Human Genome Project. So it started in 1990, initiated by United States Department of Energy, National Institute of Health. So they have a collaboration with the Wellcome Trust of United Kingdom, and other nations also joined such as France, Germany, Japan as well as China. And then, so all of them have distributed the work. Yes, there are what we can say that mainly the 46 chromosome, but one set of chromosome is enough to sequence. So ultimately what they did, so instead of 23 chromosome, yes, they have done the sequencing of 24 chromosomes. What is that? So the sequencing of 22 autosomes plus 1 X and 1 Y chromosome, plus 1 X plus 1 Y chromosome. Yes, so total is sequencing of 22 autosomes plus thus 24 chromosomes. 24 chromosomes sequencing is completed. So this is all about what we say that the history of the mainly what we can say the Human Genome Project, which started in 1990, ended in 2000, mainly completed in, it should be not 1993, 2003. Yes, completed in 2003 within a span of 13 years, and mainly the chromosome 1 was the last to be sequenced because of its huge number of genes, such as 2968 genes it is having. So this is all about the brief history as such. Yes, in the earlier class, in fact the initiation started in 1970 with the invention of mainly the electrophoresis technique. Further, the addition of other techniques such as Frederick Sanger's automated technique of sequencing also utilized, PCR also utilized. All these, yes, made the Human Genome Project to be accomplished within a well planned time or advanced 2 year advance in the whatever the time assumed. Well, so this is all about the history. Let us go ahead with what are the major applications of human genome project. So this what we study all about the mainly what we can say the history, and when we look into the mainly the such as applications, yes, enormous more applications, yes, we can find with the human genome project. So what are those important the applications as such? So once we know that the human DNA is so and so like this, the sequence of formation of the insulin, the sequence of formation of the growth hormone, once we come to know that yes, this is the DNA, this is the sequence, it is going to form particular RNA, it is going to form particular protein, the protein may be insulin, the protein may be growth hormone, the protein may be FSH, anything. So all that, yes, mainly because of DNA. So once such DNA, yes, its secret is identified and understood, that DNA we can insert it into the either plants or in the animals. So those plants or animals, when their DNA is modified by the introduction of the human DNA, and these will start producing the therapeutic proteins or medicinal proteins that is needed for the human body, and then we call it as a, yes, it is essential in the implementation of the mainly the crop or the animals, development of the transgenic animals and plants for the sake of or the benefit of the mankind. So let us understand now what are the HGP applications. Number one, it can be the crop improvement, yes. So at the time of human genome project, number of techniques have been developed, and then those techniques also utilized very well in sequencing of the number of plants also, number of animals also, as well as the human DNA also sequenced. So HGP indirectly helped in the improvement of the crops, mainly to bring about yes, the high yield, or we can say improvement of the productivity, as well as to develop the disease resistance, and crop improvement, even what we can say, yes, salinity and drought resistance. Okay, so crop improvement also, yes, introduction of the human DNA, introduction of human DNA into the crop or the plants, and plants become transgenic plants, and they started producing human therapeutic proteins. Even vaccines also can be produced, so that a watermelon or a cucumber or a carrot or a brinjal, okay, or what all these plants can have the vaccines or the therapeutic medicines. So no need to keep on taking the medicines. Yes, eat these kind of modified vegetables or the plants or the fruits, you will get the medicine. So crop improvement either by the high yield or productivity, disease resistance, salinity and drought resistance, even the content will be mainly improvement, what we can say therapeutic proteins. This is what we can say the first application. Second, health sector application in the health sector. So what is in the health sector? Yes, mainly gene studied. So particular defect understood and specific gene target medicine developed. So this field we call as pharmacogenomics. This is we call as a pharmacogenomics. So thus in the health sector also lots of what we can say the applications. As such there are 30,000 functional genes what we have, and if one gene is defected one disease will occur. That means there is a high chance of 30,000 diseases. So such kind of 30,000 diseases can be well understood by analyzing, understanding the human genes. Okay. So what we say, the information of the HGP very much useful in the health sector to produce a particular medicine, to understand a particular disease or disorder also. Then other application as such, environmental remediation. What is this environmental remediation? As I told you, HGP was not just alone, it has been carried out. Along with the HGP, people studied the genome or sequences of other plants, such as it may be Arabidopsis, it may be the rice, number of plant, yes, they studied. And certain plants also they developed. As such, they have a ability to trap the certain heavy metals in the water. And then when such plants are grown in the waterlogged area where the polluted water is flowing or going or depositing, then these plants will trap those metals, and thus they bring the bioremediation. Yes. So thus environmental remediation or bioremediation we can say. So environmental or bioremediation to reduce the pollution. So to reduce pollution. Then number 4, energy production. Yes, energy production also one of the major application. So energy production, mainly we can say that number of microbes are there in the nature. Yes, along with the HGP they studied sequencing or the DNA sequences of the number of microorganisms, that include the E. coli also, and usage of the microbes in the production of the energy. So energy production also one of the application or outcome of the human genome project. Other than this, number 5, sequencing of non-human organisms DNA. What they have done. So what are the non-human organisms as such? We will understand one by one. Those non-human organisms as such, the mice or the mouse. So mouse is a singular. Mouse DNA sequenced 99% the DNA, yes, similar to human DNA. Similar to human DNA, we say mouse sequencing. Then yeast, the unicellular fungus yeast utilized very well in number of microbiological processes. It may be the production of the beer, it may be the formation of the bread, okay, cake, many more. Yeast is very much utilized in the brewery industry also. So the yeast mainly, such as scientifically it's called as Saccharomyces cerevisiae. Yes, it's a sequence also done, studied at the time of a human genome project. And then other than that, E. coli also called as Escherichia coli, which is mainly seen in the intestine of the human. It is a symbiotic kind of organism that stay in the intestine and also helps in better digestion also. Well, E. coli. Then there is what we can say that mainly. So rhabditis In developmental biology, this organism is very much utilized. It is aquatic non-parasitic nematode. Aquatic non-parasitic nematode. You have studied Wucheria bancrofti nematode that causes elephantiasis, right? Like that. Coenorhabditis is also a nematode, but never ever harm anybody. It is the organism useful in study of the developmental stages. And then, well, Coenorhabditis. And then one more, the nematode you might have been studied, Melanogyne incognitia. So Melanogyne incognitia is mainly causing the tumors in the roots of the tobacco. But there are number of examples of the nematodes, but one is non-parasitic nematode Coenorhabditis. At the time of HGP, they have done the sequencing study of this organism also. Then Arabidopsis, a plant, Arabidopsis. Also sequencing, they studied the sequencing of the rice plant. Okay, so sequencing of these plants is helpful to develop or to improve the variety of these crops and then make them the best bioreactor. Means the medicine will be available in these animals when we make them as a transgenic Arabidopsis as well as the rice. Then Drosophila. Drosophila is also called as fruit fly. Fruit fly is extensively utilized mainly in the study of development. Okay, so in the study of embryology, yes. So Drosophila is very much utilized to study mainly mutations. So thus the sequencing of these non-human organisms also done at the time of Human Genome Project, and these are the few important applications of Human Genome Project. Well, so let us go ahead now with the further study of Human Genome Project as such. So Human Genome Project, mainly we need to understand what kind of methods have been utilized. So methodologies we need to understand. So these methodologies are very very important here, and then we will discuss about the salient features and the challenges of the Human Genome Project. So when we talk about the variety of methods have been utilized mainly to carry over this Human Genome Project, which is a massive project, very huge project. It is not so easy to accomplish this project. So they utilize lots of techniques or technologies. Let me tell you methodologies in the Human Genome Project. Methodologies used in Human Genome Project. Number one is called as ESTs. Okay, we'll discuss about that. Then there are again what we can say that mainly we can say that sequence annotation. Second method they have used. Number three, yes, the sequence annotation. Then we can say that mainly, yes, cloning of the required DNA. Yes, the method what they have used, we will discuss that also. And then, so, yes, one more important method, automated. Yes, we can say that number 4, automated sequencing. And number 5, physical mapping of genes. Yes, we are going to discuss these two also. First, let me tell you what is ESTs. So, we can say that EST refers to expressive sequence tags. So, EST refers to be expressive sequence tags. Means what? The DNA has enormous amount of sequences, but every DNA cannot form the RNA. So only particular sequences are expressed as RNA, and those expressive sequences, yes, we can say them as a mainly expressive sequence tags. Identifying them is a most important method, and then only we need not to keep on fighting with the 3 into 10 raised to 9 billions of base pairs. Use the restriction endonuclease enzymes, cut the DNA and target what is the sequence they are going to express as RNA, and study that only, because those sequences are very important for us. And then sequence annotation. So when we are studying the entire genome, so sequence annotation, they can be coding or expressive, and they can be non-coding or non-expressive. That means what? These will form mainly further, they expressed in the form of RNA, and RNA will become the form the proteins, right? So this will happen with the coding and non-coding. When you study the entire DNA, in that DNA we are going to do the sequence annotation, which are coding, expressing themselves as RNA, then they are going to form the protein. This is what we can say that sequence annotation. So some of them will never form, yes, they are considered to be as a junk DNA, because hardly 2% of the DNA of the entire human genome is going to be expressed as a RNA. Remaining 98%, yes, remain silent, will not be expressed. So we call them as a non-coding or non-expressive DNA. We need to understand that also. Then cloning of the DNA. Yes, when you isolate the DNA from the huge mass of DNA, and that DNA is very useful for you, and you want to go with a detailed study of that small fragment of DNA. It is making multiple copies of desirable DNA, of desirable DNA. This is what we say as a cloning. Yes, you can insert mainly the DNA into what we can say that making BAC, bacterial artificial chromosome, and then YAC, yeast artificial chromosome. Okay, so by using BAC and YAC, as these cells keep on multiplying the number of times, very high level multiplication capacity they have, as they multiply, they will make a multiple copy of the DNA what you inserted, and this is called as mainly the cloning of the DNA. So thus we have discussed most important what we can say mainly the cloning here, the DNA. So bacteria such as usually what they do, they will imagine here is a bacterial cell. So bacterial cell, and it has its own genomic DNA. So its DNA is there, and then the bacteria have the circular DNA, what we call them as a plasmid. So this plasmid you are taking, you are opening up this by using the restriction endonuclease, you are using or inserting what we can say that human DNA. So human DNA, yes, you are inserting. Now it become what we can say rDNA, recombinant DNA. Now you are inserting back into the bacteria. So yes, you have inserted the DNA here, and that is your desirable DNA, and this bacterial cell as keep on multiplying several times, yes, your what we can say the copy of the desirable DNA also will be, yes, will be copied. So as and when the plasmids keep on multiplying, or they keep on mainly replicating, plasmid means Yes, it is a closed circular double-stranded self-replicating DNA that's present in the bacteria or even in the yeast cell also. Yes, you modify the yeast chromosome or bacterial plasmids and you are allowing them to multiply several times. Millions and millions of cells will be found, but millions of times the DNA what you have inserted also will become duplicated, and this is called as mainly the gene cloning. So, by using bacterial artificial chromosome as well as yeast artificial chromosome. So, remaining techniques, automated sequencing and physical mapping we will discuss in the next class. Till then, take care, study well. Thank you very much.